Hello students, we are going to discuss today the practical of the effect of enzyme concentration on amylase activity. Here we have taken the requirements like first of all we have prepared 1% starch solution. We have prepared 1:20 diluted salivary amylase solution. Then we have iodine tubes already with us, and then we have done the practical by taking different enzyme concentrations. Here in this practical we have not changed the concentration of substrate starch. Starch is taken thoroughly in the same quantity, but only we have changed the concentration of enzyme, so we are able to study that what is the effect of changing enzyme concentration in increasing manner or in decreasing manner, and how it will affect the activity of amylase and what will be the effect on rate of reaction. So in doing so we have found that in 3 different sets we are going to see different results. Here first of all we have prepared 3 sets, and very first set we have prepared of very low concentration of amylase enzyme, that is 0.01 ml of enzyme. In the second reaction mixture we have taken 0.1 ml of enzyme, and in the third set of reaction mixture we have taken 0.2 ml of amylase enzyme. Now then after we carried out the entire practical for 3 times by taking different enzyme concentrations, and we have found out that reactions vary. So first of all we will try to see the reaction of the enzyme in which concentration of enzyme is very less. So here we can see very nicely that there will be very gradual development of purple color. In the initial tubes we are not seeing any type of change in the color. We have done this practical by taking out the aliquot from the reaction mixture after every 1 minute. So we have started like taking results or aliquot for 1 minute, 2 minute, 3 minute, 4 minute, 5, 6, 7, 8, 9 and 10. So accordingly we have added our aliquot, that is 0.1 ml aliquot in this iodine tube, and we have found this type of result. So in initial stage of the practical we are not getting any type of purple color formation, but in the last tubes I mean to say that in the tube number 7 and 8 we are observing the faint coloration. Okay, so especially in the tube number 7 we can see very nicely that purple color has been formed. So this particular reaction or this particular result was then after confirmed by comparing it with the blank. This blank is prepared from the starch, so we can see that this is blue color and this is purple color. Okay, so here we can find out the dextrinization time in case of less amount of enzyme. So 7 minutes is our result for enzyme concentration 0.01 ml. Then after we will going to check the result of 0.1 ml of enzyme. Here we have done the same practical, and we found out that in the last tube, that is in the tube number 4, we can see that purple color has been formed, and it was compared with the blank also. So here we can see very nicely that this is dextrinization and this is our blank for starch. Okay, so it is not having the blue color, but it is having purple color. And then we can also have the look on all the 4 tubes of 4 different minutes. Okay, so here we can see very nicely, this is our first minute result, this is second minute, third minute and fourth minute. We can see very nicely that dextrinization is done. Okay, so in case of moderate amount of enzyme, we are getting moderate amount of dextrinization. And lastly, we will see about the concentration of enzyme, that is 0.2 ml, that is double than 0.1 ml. Now here we have found the result directly on the first tube. Okay, directly in the first tube we are getting the dextrinization. So that was also compared with the result. Okay, so you can see very nicely that dextrinization is already taken place. If you want to see it more clearly, I will show it to you. Here we can see that there is a very clear purple color formation. So now at the end of this practical, what could we conclude? We could conclude that as we increase the amount of enzyme molecules, there will be increase in the rate of reaction. Okay, so in case of less amount of enzyme, we have seen that so much of the time is required for the dextrinization to happen. So it says that some enzyme molecules are there which are already in the reaction, and starch molecules are also there. Okay, and there was some time which was required to have the binding between both of them. So that is why we require more amount of time for getting the dextrinization. But in case of 0.1 ml of enzyme, what we have seen that only 4 minutes, after 4 minutes we have seen the dextrinization already done. So it means that now the stage will come where there will not be any substrate starch or any starch molecule which will be not bound with the enzyme molecule. So now onwards, if you increase the concentration of enzyme, there won't be any effect on the rate of reaction, it will be the same one. So here we can see in case of higher amount of enzyme, the dextrinization is done only in the first minute. Okay, that is not a single second. Okay, this was immediate reading, that is 0 hour reading. You can see that dextrinization is done. So what it indicates? It indicates that now onwards the starch molecules are not available in the reaction mixture. So if you go on adding the enzyme molecules, there won't be any change in the rate of reaction. So here we can conclude from this practical that as we can increase the enzyme concentration, there will be increase in the rate of reaction. But when there will be saturation of substrate molecules, now no more substrate molecule is available to bind with the enzyme molecule, the reaction will not happen and we will not have any increase in the rate of reaction. So I hope that you have understood the practical thoroughly. Thank you very much.